Hello everyone. Firstly, before we start, I would like to check whether everyone is able to see me and hear me. Do you hear me? Okay. Guys, do you hear me? Yes?
Yes.
Perfect. Thank you very
Yes.
much. Okay, so very well. Also, you are able to see me. Second point.
Yes, I can.
Perfect. And also, are you able to see a slide where is stated Intro to Biology Studies?
Yes.
Perfect. Thank you very much.
No, I can't hear your voice.
You can't hear my voice? No? Okay. What about the others? Sora, you have a problem with hearing my voice? And what about the others? Misato? Okay?
Okay.
Perfect. Yuta? Clear?
Okay.
Perfect. Now?
Yes.
Okay, I can hear you.
Okay, now what about you? Can you hear me and see me? Okay, now it's maybe Aoi? Okay, perfect. Satsuki? Good?
Okay.
Perfect. Mona? Perfect. Soko?
I can hear you.
Everything good, yeah?
Yes.
Okay. Yuiko? Okay, perfect. And Yuki, I cannot see you, Yuki. Okay, perfect. Can you? Okay, so now everyone is able to see me, hear me, and also see the slides, right? Okay. There is some echo. Okay, okay. Perfect, guys. So hopefully now everything works. And I would like to start today's class with a little introduction about me, about you, and also about the biology and the classes we are going to have together. There are some slides prepared for you. Nevertheless, please let me introduce myself firstly. So hello, and my name is Samo Stefanko, but you can call me Samo. Um, I come from Slovakia, and my background is general medicine. I was studying general medicine at Charles University in Prague, in Czech Republic, first faculty. So at first faculty of Charles University, I was studying, and I ended my studies back in 2021. Since then, I'm now currently my job position or my full-time position is a online tutor, and amongst my interests belong soccer, literature, hiking, and yeah, many other things. Also, I would like to maybe mention that one of my specialties belong psychotherapy. I'm attending now a psychotherapy course. Currently, I'm in the second year of my studies of psychotherapy. So, yes. Um, Perfect. Now, if you do not have any questions, or if you have any questions towards me or towards my background, you can ask. But I would like to also sometimes I cannot hear your voice. Sora, is it working now? I cannot see Sora and Yuiko. Are there any difficulties? Sora. Okay. Mm, I will just write answer to Sora if she's not here. Sora, try to refresh. Okay. Can I ask you maybe three basic questions. Three basic questions about we can start it. You will tell me just your name or nickname, any preferred nickname, or how would you like to be called here in the classes. Your background, it will be very interesting. Your background and maybe interests. And one additional question would be, what kind of doctor would you like to become in the future? So, if it's okay for you, it would be very great to hear a few words from you at the beginning. So, Misato, maybe can you start, please?
Yes.
Perfect. And please
Mm,
correct me, everyone, if I'm pronouncing your names incorrectly, okay? So I will try to do my best. So Misato, am I pronouncing it right?
yes.
Perfect.
My name. My
So
name is
maybe just about the background, mm, about your educational background, and what kind of doctor would you like to become in the future? Those two things.
I would like to be a doctor. I would like to be surgeon.
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 Okay.
Yes.
Maybe can you put a mic closer to your mouth because I was struggling to hear you. Maybe now?
Okay.
Yeah. Yes, yes, that
Yes.
works better. Surgeon. Okay. And what about the background? Do you have uh, any experiences with biology?
Yes.
Yes.
I experienced biology at secondary school.
Okay. Perfect. Perfect. Okay, great. Thank you very much, Misato. Yuta, what about you?
I want to become a physician in internal medicine.
Internal medicine. Perfect. Okay, internal medicine. Yuta, am I pronouncing your name right?
Uh, Utah.
Yuta. Perfect. Thank you. Okay, internal medicine. Have you studied biology before?
No,
No.
but
Okay.
I study by myself.
Okay. Okay. So there is some knowledge. Okay. Okay. Perfect. Thank you very much, Yuta.
Thank
Aoi,
you.
what about you?
Yes.
Okay. So what kind of doctor would you like to become?
What kind of doctor? I want to be pediatrician.
Pediatrician. Okay. Interesting. Pediatrician. Do you have any background regarding biology?
No background.
No background. Okay. hmm Thank you, Aoi. Mona, what about you?
I want to I want to become pediatrician too.
Okay. So already two pediatricians. hmm Okay. What about the background?
Yes, I studied biology at the school.
hmm Okay. Perfect. So there is biology background um, from a school. Perfect. Thank you very much, Mona. Shoko or Soko?
Shoko. Yes.
Shoko.
Shoko.
Okay.
Yes.
Thank you.
Correct.
Okay. Shoko, what about you? What kind of doctor
Uh,
would you like to become?
I would like to be an emergency doctor.
Mm-hmm. Okay.
Yes.
And
And
biology background?
I studied biology in high school.
Perfect. Okay. Mm-hmm. Thank you very much, Shoko. Yuiko.
Yes.
And am I pronouncing right?
Yes, Yuiko.
Yuiko. Thank you, Yuiko. Um, what about your dreams? What kind of doctor would you like to become?
I don't think what kind of doctor
Sorry,
yet.
can you repeat? Didn't hear very well.
So I don't think what kind of doctor yet.
You don't know yet. Okay. Okay.
Yes.
There are plenty of time in front of you, so Definitely, you don't need to have a specific specialization now. Perfect. What about the background? What about the background? Have you studied biology before?
Yes, I studied biology in senior high school.
Perfect.
Yes.
Okay, okay. So there is a biology background. That's great. That's great. Thank you, Yuiko. Yuki,
Yes.
what about you? What kind of doctor?
I would like to be a brain surgery.
Brain surgery. Okay.
Yes.
So neurosurgeon, brain surgery or neurosurgeon. Perfect. What about biology? Do you have any background?
Yes, I did in high school.
hmm Okay, so there is a biology background as well. Thank you, Yuki. Now,
Yes,
so what about you?
I would like to be a ER doctor.
ER doctor. Okay. And what about biology?
Yes, I studied biology when I was a high school student.
Perfect. Okay. Okay, guys. Thank you now. Sora?
Yes,
What about you?
I want to be orthopedics in the future.
Orthopedics?
Yes,
Perfect. Okay. hmm And what about the background?
yes, I have. I studied biology at school
hmm
in senior high school about 1 year.
Okay, so there is some. Thank you very much. Thank you very much, Sora. Last but not least, Satsuki. You are muted, Satsuki. You have to click on the mic up there if you want to unmute yourself. Perfect.
Yes,
Yes.
uh, I want to be a heart surgeon.
Heart surgeon. Okay, heart surgeon. And what about the background regarding biology?
Yes, I studied biology when I was a high school student.
Perfect. Okay. Okay, guys, thank you very much. I've noted something about you. Thank you very much that also you guided me regarding the pronunciation. And please, any in the future, when I will be pronouncing your names incorrect, please correct me. Okay? I will try my best, but please correct me. Uh, okay. Let's get to the next point. Okay, so let me use my pointer. So let us get to the third point, session structure. What does it mean partially? Flipped classroom, and what about the form up score and summary activities? Okay, guys, I understand. Here we now we are in the beginning of the summer, and I would like to create, or I would like to structure the sessions in more interesting way for you, that it won't be just covering the things theoretically. What does it mean partially flipped classroom? Basically, flipped classroom is a type of education or type of classrooms when the students are always active and when they are providing the others with the information. It's something like peer-to-peer learning. So basically, for example, one of you will cover something and the other one will cover something else. And you will have a discussion and we will discuss that. Not the whole class will be like this, but there will be some activities during the whole course when we will have activities in this way as a part of our sessions. Why I think it's important, because it's way of active learning processes. When you are actively doing something, when you are collaborating, searching for information, discussing the things together, it's it's more likely that it will become part of your long-term memory in the hippocampus. Basically, it will stay in your head longer, the information, comparing when you are just passively learning things. So basically, when you are just hearing or watching something. So I would like to have mm, complex lectures or sessions with you. And now that brings us to the whole structure. So warm-ups. Each session will start with a little warm-up activities. Mostly, I think it will be quizzes from previous topics we covered, and also it will be uh, based or structured like the admission questions. So there will be some question, and there will be options, and we will discuss each of them. Why is it correct? Why it is incorrect? And also this is kind of an active learning process, because we will have a discussion and we will understand why some options are not correct, why the one option is correct. Okay, so basically this will be a quiz and nothing struggling, I would say. The core of a session will be usually some theoretical parts we have to cover together. We will discuss many things regarding the biology, and also some clinical parts. So basically there will be some I would like to connect the theoretical part with some clinical knowledge, or with the understanding why we are learning this and this. Okay, and what is the necessity for the future, for you as a future doctors, why you are learning this. So basically, this is a clinical part. And summary activity, basically there is some visualization activities. So for example, when we are dealing with the biology of a man, there will be plenty of things, for example, which are related to anatomy or histology. So visualization activities are very good for example, also for memorization, like drawing or watching some short videos. I will upload many videos to the Google Classroom for you as well. Nevertheless, do not want to I do not want to build those sessions that you have to spend hours to be prepared in advance for the session. So this is not our case, okay? We will try to do everything here. So we will be like 100% of the time active. We will be studying, we will be preparing, discussing, and so on, and so on, and so on. So you will have to like do really just a little, maybe as a homework. So maybe some watch a video, short video, and that will be everything. Okay, so most of the time I would like to spend the time here with you. So when you are done with the class, you will gain some knowledge, but also will not have to additionally study in your free time. Okay, and also the last point here, your feedback. After when we close one chapter of biology and the other, I would like to hear your feedback uh, towards regarding the session we had. So how did you like the structure? What was good? Whether you like this activity or not? What would you improve? How would you maybe modify that? Or maybe would you like to keep this activity or this structure also for upcoming topics? So your feedback and your opinion is very important also for me, and also for the whole atmosphere here. um, yes, so basically that was regarding the structure of the sessions. Do you have any questions so far? No? Is everything is okay so far? Yes.
Yes.
Okay. Okay, guys. Just maybe to not confuse you, I will be operate on two screens basically. So here I see you on this screen. Okay, when I'm looking here, I'm just seeing you, and here when I'm looking here, I'm seeing the presentation. So let me just start that. Okay, 1 second. Okay, perfect. So here we have a presentation. Okay. Okay. So, guys, tips for studying. I also mentioned at the beginning Timotej Pazitny, or Timo. I think you already met him, right? No? Am I right? He's my colleague here, and his insights regarding the tips for studying, I think, are very useful, very beneficial. So that's why I was inspired by him. So I would like to also give him a credit for those points, guys. So tips for studying. Let's have a look on the first one. Make a plan. So every every successful entrepreneur builds their business on a business plan. So why shouldn't we help ourselves in the same way? Choose subjects and topics that are a priority for you and estimate how much time they will take. Do not hesitate to write such a plan on paper or in a calendar. Okay, those tips I think are not relevant yet for you, like for this summer course, but those tips are very essential for your medicine, for your medicine studies, and also for preparation for admission. um, Guys, I think it's very important to be strategic regarding the biology or also chemistry and physics studying, because there is a incredible amount of knowledge and information you have to keep in your head. And as you know, the repetition is a mother of knowledge, so you will have to repeat many things many times. So that's why those points here are very important, very essential for your future. Okay, for now, we will do everything together. You don't have to, I don't think you will have to spend a lot of time regarding the studies during this summer, but take these points as a guide for your future, because Timo and also me, also Petrana, we were all all of us were studying medicine. All of us understand what does it mean when student is struggling with something. So those points are essential for your future yourselves. Okay, so make a plan is one first point. Set aside time. When you want to save money successfully, you set aside reasonable amount of money from each paycheck and only spend the rest. So do the same with your time. Set aside time dedicated to studying and stick to it. Then you won't feel stressed during your leisure activities and you won't feel like learning is stealing something from you. Okay? Very, I would say, overlapping with the first plan. Make a plan, set aside time. So basically you try to create a space or create a time range during your day, during your week, during your month, when you are going to study and how much will you study. If you planned it in advance, it will be also your advantage. Okay, provide plenty of light. So yes, basically it's about the brain activity. If you are really in a dark room, also your concentration is really decreasing. So that's the point basically. Seek silence, very similar. Student can be very easily disturbed by many factors around if there is some kind of noise, or other people are doing something else as studying. For example, they are playing video games and stuff, and your concentration have like leakage. So you just have a look on that and you lose your concentration of studying. So yes, engage your senses. Engage your senses is basically mean you don't have to study only from a text. We live in the 21st century already, and we started third decade. So basically there are plenty of videos, plenty of interactive things, plenty of other possibilities how you can study, not just only from a text. Okay, so basically that. And that's also very similar to utilize technology. And this is a very important point. Don't leave any word misunderstood. If you are studying something, or also when you will be not familiar with any words during our classes, please stop, raise a hand, and I would like to know what does this word mean. I'm not familiar with that, okay? Because if you do not understand it now, it's very probable that also in the future you will not be familiar. So yes, yes. Just don't hesitate to ask questions basically during our classes. Get rid of what you already know. This is a point maybe for already the last phase of your studying before the admission and also before the exams, when you will not have already like plenty of time, and you have to be again strategic. So if there are topics you are already familiar with and you know them, you don't have to waste the time with repeating them, and maybe try to focus on something you are not good as good as you would like to be, and focus on them. Okay, but okay, we are not just you will not be just focused on 100% of your time just focus on text, because you can easily be frustrated and you can easily be desperate. What find very useful, or what I found very useful during my medicine studies, was to find a study partner. You can really he or she can really help you with the others, and studying in a pairs can provide a new perspective on the topics. Uh, plus additional ad, other thing to mention, you are not alone in this. Competitiveness may motivate you better prepare for such meetings. Additionally, topics discussed together are more likely to be remembered. Again, it will be in the long-term memory. So very, very useful thing when you have a studying partner for that. Relax while moving. So basically, try to do any kind of sport, and don't read everything. Don't read everything in once. Okay, do chapters, do it one by one. Okay, do it digestible for you. Also reward yourself. So if you are a sporting person, or I don't know, maybe you prefer to move, um, tell yourself, okay, today I'm going to read a cell. For example, the chapter of cell have like 20 pages. So after 10 pages, I will be going out to have a run, or I don't know, if you prefer to play video games, I will play for 30 minutes video game, and then I will get back to the cell topic and I will finish it. You will be satisfied and won't be frustrated, and it trust me, it's very easily to become frustrated during the medicine studies. So this is basically the fourth point: reward yourself. Check your ambitions is basically um, like I will tell it on the example when you are already in the first year of medicine, or you will be now during your year focusing on the admission. For most of the school year, our approach to preparation is mostly passive, but the exams and the final exams are very important, and stress comes very easily, and for some people the tendency to catch up on everything uh, is like dive headfirst into it follows. So high demands and goals that you or your family set for yourself can be a good motivation, but also uh, it's important to mention, try not to overdo the things. Okay, so some kind of balance to find in between studying and also your personal life. So try to handle the balance. It will be very hard from the beginning, but it's doable. Let's do it this way. one last point regarding the studying tips, and we will get to the topic. So, journalism method. I don't know whether you are familiar with that, but I would really like to mention this method, and I think it's very beneficial. Basically, you can do it at the beginning from this very simple or in this very simple way. So you will kept your journal. You will um, mention a date of today, and you will answer two basic and simple questions. What do you think you did well today, and what can be improved? So you will be studying something, and you can always after I don't know of after a day of preparing, you can open this journal and answer those two questions. What was struggling point for you maybe? Can be also other possibility how to structure the question, what can be improved. Okay, rate your performance on a scale 1 to 10. You will do this and you will not look at it for a week or two. After a week or two, you will get back to your notes, to your own notes, and you will see whether there is a progress or not. Okay, whether you are um, improving your flaws, or checking, yes, I'm doing progress. And if you are able to see it on a paper after a week or two, yes, I'm doing progress, is really a rewarding feeling. So this is a thing um, I would like to mention for you before we start. Okay, any questions so far? No? Okay. Let's get to the technicalities. Neuro and Google Classroom. Now I would like to ask all of you whether have you already were on Google Classroom link? Yes? All of you were already there?
Yes.
So there are few posts from me already. Few posts from me. So basically, I asked you whether you can comment a first post which was posted on 3rd of July. And I see there are 7 comments below, and also then I uploaded one PDF book and few videos which will be relevant for today. Okay. Any questions regarding Google Classroom, guys? It's, I would say, very similar to Facebook. Basically, I will post the things there. Most of the time, I will try to maybe post some videos or some articles for you, but mostly or most of the knowledge or information I will try to provide you here during the classes. So really just short videos for you, maybe for as a recapitulation of what we already discussed, what we already covered, or maybe something what we are going to cover tomorrow or on the upcoming day, basically. Okay, and this is a new row. This is a platform when we are where we are going to have our classes. Um, Basically, I'm not sure whether you already had a classes last week. Did you? Did you have? Yes, you had already classes. However, it was on a different platform or it was here in the new row. Misato, you were nodding. Do you hear me? So it was in Euro here?
Yes.
Yes, okay. So you are already familiar with the Neuro. Guys, there will be, as I mentioned already, some different types of activities. What I really find, and also most of the students find very interesting, that for some activities you will be breaked out in smaller classes. So basically we will be kept here in the neuro, but you will collaborate in a smaller groups, maybe or individually or in a pair or a group of 3 people together on some task. Okay, basically, so that's the thing which is one of the many features here in the neuro. I see you are nodding with the heads, so you are, I guess, familiar already with breakouts. Perfect. That's the great then. Okay, guys, so that's basically it. One more thing additionally, I know that, in other words, I'm already recording the session. Are you okay, everyone, with that? That we are recording the session, so in order you will be able to see the recordings even after the session is done. Is everything okay for that? For you that the sessions are recorded, because it's one of the requirements from the management, let's say. Yes? Clear?
Yes.
Okay, perfect. Great. Thank you very much. Um, what I think it's the best way how to upload the sessions, or where to upload the session, is a YouTube channel. Okay, it will be private YouTube channel, so not everyone will be able to see it. However, I will upload the sessions there. After discussion with my colleagues, we found it as the best way how to do it. So basically, I don't know. No, basically the recordings will be uploaded there. So I will always, when the recording is uploaded, I will again put the link with the recordings on the Google Classroom, and it's up to you when and where would you like to watch it. Okay, so basically that's it. So it will be on the YouTube, on our private channel. Okay, guys, so I think it's everything, and let's start. Do you have any questions before we dive into our first part and first chapter of biology, which will be a cell? Anything what comes to your mind? Nothing so far? Perfect. Okay, so let me just change a presentation. Mhm. Okay. Okay. So, guys, we have 13 minutes before the first break. Yes, maybe one other question regarding the structure. Would you like to have a session 2 times 50 minutes, or in one time 100 minutes? We will do a poll, okay? So uh, the it will be question yes or no. Yes, I want a break. No, I don't want a break. Okay? So here a poll. Yes, I want a break. No, I do not want a break. Okay, 2 more people. Okay. So it was close. However, no break. Okay, so for today we will have no break. Tomorrow again I will ask. Maybe your mood will change. Sometimes the break is necessary for topics, sometimes not. Okay, but today we are not having a break. So guys, it was democratical vote. I cannot change it. So sorry for those who wanted a break. However, the majority of your group did not want it. So we will continue with our today's topic, which is biology of the cell. Okay, guys. So here comes the first slide. On the left side, we are having a prokaryotic cell. On the right side is eukaryotic cell. The question anyone can answer. Please do not raise a hand, just unmute yourself and share whatever you want. What are the differences, or how is, let's make it better, how is a prokaryotic cell different from eukaryotic cell? Anyone?
Prokaryotic cells don't have nuclear.
Perfect. Okay. So yes, prokaryotic cells doesn't have a nucleus. Perfect. What else comes to your mind? If you know. If you know. Okay, so we can say in general that prokaryotic cells are smaller, okay, are smaller than eukaryotic cells. Okay, so let's say a typical bacterium is a spheric, and the range of the length or diameter is from 1 to 2 micrometers. Okay, micrometers, 1 to 2 micrometers. So this is basically a range of prokaryotic cells. We differentiate a structure of prokaryotic cell into prokaryotic prokaryotic nucleus or DNA, prokaryotic DNA, cytoplasm, and plasma membrane. However, as you said, Shoko, exactly, there is no nuclear envelope around the prokaryotic DNA. So this is also a very big difference comparing to eukaryotic cell. If I ask you, do you have any idea uh, where does the eukaryotic term come from? What does it mean, eukaryotic? Maybe. Any ideas?
I don't understand that
Yes,
question.
okay. Yes, okay. I will structure the question differently. Um, the eukaryotic, as a term itself, has its meaning. It's derived from the meaning. What does it mean? What I want to say basically is that karyotic basically refers to karyo or to nucleus, and U or EU is basically what is something true or owned by yourself. So you have the eukaryotic cells have their own or true nucleus with everything what nucleus has to have. So nuclear envelope, and is differentiated with the membrane from the surrounding basically. So eukaryotic cell basically means that this cell has a true nucleus. So that was it. Okay, so that brings us back to the structure of the prokaryotic cell, that we do not have nuclear membrane there. Okay, so yes. Other very important thing to mention is that the basic prokaryotic nucleus is not anyhow differentiated to the cytoplasm, what we already said, and also the the prokaryotic cell does not divide by mitosis. Okay, um, unlike eukaryotes, prokaryotes undergo a type of cell division known as binary fission. It's somehow similar to mitosis, but definitely it's not a mitosis. So we can say that this is another differences between prokaryotic cell and eukaryotic cell. They are not dividing in the same way by mitosis. Eukaryotic cells does, prokaryotic cells does not. We will cover mitosis. For those who are not familiar with that so far, it's okay. We will cover mitosis in the following days. Basically, it's the way how the eukaryotic cells divide. Okay, we have one cell, one eukaryotic cell, and the mitosis is the process how from one eukaryotic cells we get two. Okay, so this is mitosis. And when we are talking about division or dividing cells in a prokaryotic cells, the process is a little bit different. We call it binary fission. It's not so important for you and for your admissions, but this is one of the differences between those two cells. Okay, so far we mentioned what we have mentioned so far. Yuki.
Yes.
So what we have mentioned so far is the differences.
Differences.
We have mentioned the size, that the prokaryotic cells are smaller. Yes? We have mentioned also difference that nucleus or nucleus in the prokaryotic cell is different, that it doesn't have a nuclear membrane. Okay? So nucleus is also different in prokaryotic cells. Also, what is different, the third thing, is that a prokaryotic cell divides differently comparing to eukaryotic cell. Okay, so also the cell division is different. Okay, so far 3 points we mentioned. Yes? Thank you.
Yes.
Perfect. Guys, if we are going too fast, stop me. If you have any question, anything you don't understand, raise hand or just voice it out. We will discuss that. Okay? I understand you don't have to know those topics. It's summer and so on. Nevertheless, we have to cover plenty of things, so we have to have a pace. Nevertheless, no matter what, if there is something you don't understand and something you want to repeat, something you want to clarify, please ask. Okay? Because if no one is asking, I do not know then that you are struggling maybe. Okay? Perfect. Perfect. Okay. So what else we have to mention regarding the prokaryotic cell? The nucleoid or bacterial chromosome consists of one single molecule of double-stranded DNA. Okay, so also prokaryotic cells have a double-stranded DNA. I will draw it. So basically, DNA, maybe you already saw this picture. Okay, so this means it's double-stranded. Okay, so we have some bases, some sugars there, and double-stranded DNA, and it's just wrapped there inside the cytoplasm without any membrane around. Okay, so double-stranded DNA is a similarity. What is difference is that there is no membrane, as we already mentioned a few times. What is necessary to mention that in prokaryotes, DNA is circular. Okay, so let me just finish the drawing. So it's like this basically. Okay, so it's in a circle. Okay, it creates a circle, this double-stranded DNA in prokaryotic cells. However, if you are able maybe to zoom in, please zoom in to the prokaryotic cell, and what we are able to see there on the picture, we are able to see nucleoid circular DNA in the middle, the reddish center of a prokaryotic cell. Are you able to see it, right?
Yes.
Okay. What are do you see also the other very much smaller reddish points, which are plasmids? Do you see them? Plasmids. If you zoom into the picture, where you have to really zoom in, but they are there here. Do you see them? Plasmids. If you don't know how to zoom in, guys, on the right side of the neuro there is plus and minus. On the right, in the right corner, down on the right side is a plus and minus, and you can zoom in. Do you see those plasmids here?
Yes.
Perfect. Do you have any idea what it can be? Any ideas? It's also a DNA. It's also a DNA. And why is it important? Those DNAs, or let's say fractures of DNAs we call plasmids, are not essential for prokaryotic cell itself. But what is important? Why the prokaryotic cell still keeps it is that it keeps, or this plasmid keeps, an information, or let's say like additional information, uh, such as, for example, antimicrobial resistance. So if you have bacteria and you have a bacteria which are able to resist some type of um, uh, antibacterial treatment, the information how they do that is kept here in the plasmids. And when they are dividing, or let's say when they want those bacteria transfer, or when they intend to transfer this information towards other bacteria, and in order to share the antimicrobial resistance and to create whole like plenty of other bacteria to be resistant against the antibacterium antibacterial treatment, they use plasmids basically. Okay, we will get there how they do that, but basically they just in a really simple way, they come together, they use a pili, which are here, pili, you are able to see, and with them they send the plasmid to another prokaryotic cell, and the other prokaryotic cell now has an information how to be resistant to antibacterial treatment, basically. In a simple term, this is how they use it. So plasmids, let's get back what the plasmids are just little fragments of DNA which are kept in the cytoplasm, They are not essential for bacteria, but they can keep, or they are keeping important or additional information which can be very useful, for example, in evolution of bacteria. Yes? So, perfect. Okay. Okay. So what else to mention? Let me check also my notes here. And yes. Okay, what else to mention? So we already mentioned pilis, and there are different types of pilis. However, there are what is necessary to mention is that those pilis are also a sexual pilosis. Basically, those prokaryotic cells are having sex with pilies. However, this sex is in order to transfer the information we were just talking about, for example, antimicrobial resistance is a great example. Okay, so bacteria are having sex in order to create more resistant bacteria. Okay, so via pili, by those pili, they transfer those plasmids via sex to the other bacteria, and the whole then the whole group of bacteria become resistant. Okay. Okay, so we discussed pili, we discussed nucleoid, cytoplasm. We will get there. Okay, what about bacterial flagellum? Here we are able to see this thing. Any ideas, guys? What it can be?
They use it in order to moving.
Perfect. So it's basically a motion organ of bacteria or prokaryotes. So in order to move or to transport, in order to be, to be mobile, they use flagellum. Okay, so flagellum is a motion organ based on proteins. Yes. Okay. Then we have a cell wall and plasma membrane. Plasma membrane, we will get there later. What do you think, guys, the capsule is? Capsule. Just ideas. You are able to see it on the picture. You see where the capsule arrow is pointing at. So whatever comes to your mind, please just share. Yuta, what do you think capsule is? Yuta, do you hear me? Okay, Yuta, do you hear me? Because you are muted and I can't hear you. Is it working? Or if it's not working on your side, maybe try to just refresh website and you will get there. Meanwhile, Sora, what do you think?
Sorry, one more time, please. I didn't understand your questions.
Okay. So capsule. What do you think the capsule is? If you just zoom in again on a prokaryotic cell, and here we have a capsule. What do you think? You don't have to be right, but just what do you think it can be?
Capsule. Okay.
So it's on the. Mhm.
I didn't know, but I think protect
Yes,
about
perfect.
protection.
Yes, exactly. So basically, it's a shell on many bacterias. Not all of prokaryotes uh, do have a capsule, but many do. And as you said, it's also for protection. So the consistency is more gelatinous, and it's like imagine it like a shell, okay? Like a shell. So basically, yes, it have a protective function. So very good, Sora. Yes. So what about then cell wall, guys? What do you think the cell wall is? Now,
Keep the shape.
Yes, also. Very good. Cell wall. So try to keep the shape. It's a solid structure outside the plasma membrane, and yes, so it's just let's imagine it like we have one layer, or the most outside layer is capsule, and just below it is a cell wall. Capsule is not in each of prokaryotes, but cell wall is. Okay, each prokaryotes has a cell wall. What about the plasma membrane? Plasma membrane. It's one layer below. We are able to see it and as a green layer on this picture. Guys, there is some echo, like something is ringing. I don't know where.
Sorry, it's maybe
Okay,
my.
maybe you can just mute yourself and yes,
Yes.
up there is the mic. Click on the mic. Perfect, perfect. Okay, and when you want to share something or answer, please just unmute yourself. But when you are silent, just mute yourself because there is an echo. Satsuki, what do you think plasma membrane is? Just guess. If you don't know, just guess. Do you hear me, Satsuki?
Yes.
Okay, perfect. Just guess. Do you think plasma membrane is something what is also in eukaryotes? If you just check and zoom out, you are able to see, sorry, there is my drawing, my bad. My bad. Okay, so we are able to see that plasma membrane is in prokaryotic cell and also in eukaryotic cell. So yes, plasma membrane is a component or organelle of cells for both types, for prokaryotic and also for eukaryotic. So yes, that's clear? Do you understand me, Satsuki?
Sorry. No,
No?
I don't know.
Okay, the question was whether or if plasma membrane is also in prokaryotic cell and also in eukaryotic cell. And the answer is yes, because plasma membrane, as we are able to see, is present also in eukaryotic cell. Do you follow my pointer now? Do you follow? Yes, perfect. And you are able to see it here. And if you zoom in a little, yes, plasma membrane is also here. So we are able to see that plasma membrane is something essential for both types of cell. Very good. Okay. We will discuss plasma membrane in more detail later on at the end of this or in the middle of this chapter. Nevertheless, yes. What else? What else to mention regarding? Okay, I think we discussed everything, and other things like ribosomes and cytoplasm we will discuss later as a part of eukaryotic cell. You don't have to know it yet, but you will know it very soon. Okay, so here we have one more picture which is zoomed in, which is much more bigger. So guys, what I would like to maybe again mention to you as a studying tip, try to draw a lot during medicine. I maybe you don't have to draw things nicely. Don't have to be a masterpiece, but many things, when you are able to draw them by yourself, it's one of the best way how to check whether you understand the things. Okay, for example, anatomy, histology in the future, also like, or these kind of biologies. Always when you are able to see a picture, try to, if it's useful picture, try to draw it by yourself. And if you are able to draw it by yourself, you know it, you definitely know it. And this will apply definitely during your first year of medicine. I know it's far from now for you, but if you are willing to pass, just do a note and try to remember that during the first year you will have to draw a lot, but it will be very helpful. So even for those topics like cells, try to draw a really simple schemes, simple drawings with each of the organelles, and try to draw them and check yourself. Okay, so here we have a plasmid. What is a plasmid? What we discussed about the plasmid, and you are able to draw it, and you say, oh yeah, plasmid is something what is like a fragment of DNA because it's close to DNA. It's in the same color, and yes, I remember that thing about the antimicrobial resistance, and you already know the thing, only because you draw it. Okay, so is also one of the hints for studying for future, because even though guys we are having now you are having a summer course which is very good, very great. I think it shows that you are willing and you are putting an effort in. During your studies, most of the time it will be a self-study, especially in the medicine. If you are going to attend if you are going to or willing to go to Czech Republic or Hungary, or somewhere else, most of the time, even though you are going to attend all of the classes, all of the presentations, most of the time it will be a self-study, your self-preparation. And if you have a good tools and good tactic, strategy basically, um, how you are going to learn it, it can really tremendously be more efficient for you, how you will be facing really challenging topics. So sorry for that. Nevertheless, I think it's just a useful hint. Sorry for again bringing some new tool how to study, but I find drawing as one of the best way how you can learn things. Okay, so here we have two pictures, one table, one comparison picture. Basically, on the left side, here we have E. coli. It's a bacteria, Escherichia coli, in a full name, and it's in the range of prokaryotic cells. So we are able to see it's 2 micrometers in the diameter. When you are comparing Escherichia coli or prokaryotic cell in this case to red blood cell or lymphocyte, you see the size difference. Okay, so it's much more bigger. Red blood cell is already an eukaryotic cell. Okay, so it's a cell which is very typical, for example, for animals or for humans. Yes, the same as the lymphocytes. You see on the first glance, you see the difference in the size. So red blood cells and lymphocytes are from 6 to 9 micrometers. So from 3 to around 3 times bigger than prokaryotic cell. This scheme here um, represents the differences between prokaryotes and eukaryotes on the left side. So yes, Bacteria or archaea belongs to prokaryotes, and organisms such as fungi, plants, or animals belongs to eukaryotes. Okay, so eukaryotes can be in a singular or multicellular form. So, what do you think, guys? Humans, best example. What do you think? Are humans single or multicellular organism? The question is. Are
Multicellular organism.
perfect, yes. Okay, so for example, Escherichia coli or the bacteria from here is a single cell organism or bacteria, or the form of life is a single celled form of life. Nevertheless, as Shoko said exactly, humans, animals, more complex forms of livings are multicellular living forms. So that's the reason why our cells are differentiated. So what I mean by differentiation, sorry, that's my dog. 1 second. Gordon, miesto. Sorry, my dog was barking. Um, what I mean by differentiation of the cells. Um, we have plenty of organs inside of our body. We have heart, we have brain, we have bones, we have liver, we have lungs, and so on, and so on, and so on, and so on. Each of those cells, for example, the cells of lungs for breathing, their differentiation and their functioning is totally different to cells, for example, in the liver. Okay, but still, even the cells of lungs and the cells of livers belongs to the cell of human, and both of them are eukaryotic cell. Even though they are differentiated or specialized differently. Okay, both of them are eukaryotic. So that's the that's what we can understand after that multicellular organism. Okay, so there are plenty of cells. Multi means like plenty or a lot of cells, which are differentiated and which will be highly specialized in most cases. For example, when we are talking about humans. Okay, understood so far? Perfect. Mhm. Okay. Organelles, cytoskeleton, cell division. We already mentioned cell division, but we will get there. Organelles are missing also as cytoskeleton is not present in prokaryotes. Okay, nevertheless, it's essential part for eukaryotic cells. Cytoskeleton and organelles, and also such as cell division, and we will get there very shortly. Nevertheless, yes, maybe today we will have time to start with the organelles and their differences. Okay, uh, so there is none in prokaryotes, and presented, they are present in eukaryotic cells, organelles, cytoskeleton, and cell division is more complicated comparing to prokaryotes. What about DNA? DNA we mentioned that it's smaller and is circular. Okay, so please, guys, this is a very important thing to note that the DNA is circular in prokaryotes. Um, I saw many times that there was a question in admission tests or admission questions like this, okay, regarding the DNA of prokaryotes. And there are no intros and plasmids. We'll get to intros as well as to many other things, but basically, guys, imagine it like I will draw a line, okay, like this, and basically imagine that this is a DNA, okay, whole line, and the red is intron, and the black one is extron, and the intron uh, is, let's say, how to explain it very simple, only for reading. It does not have um, transcription. Tra tra it is not transcripted. We will get there what is transcription, but basically, what is maybe so far essential for you to know is that there is a when we are dealing with the DNA, there are basically two parts regarding the reading: intron and extrons. And introns are something what makes uh, our DNA more complicating, and something like introns does not exist in prokaryotes. Okay, so again, prokaryotes are more simple type of living organisms. Okay, so basically those are introns. But even though we will discuss introns shortly, introns and extrons are topics for second and third year in a medicine. Okay, so for you really far from now, but you can have a knowledge what it can mean basically. Okay. Yes. So next point is RNA synthesis and maturation. It's complicated in a eukaryotic cell. We will get there. So basically, we start with the transcription. Yes, transcription in a nucleus, and then we go to the cytoplasm. There is a translation. We will get there. And yes, in a prokaryotic cells, it's again simple. Protein synthesis and maturation. It's in the cytoplasm and the rough endoplasmic reticulum. So again, it's more complicated, and metabolism is also one of the factors which is different in both. Metabolism in prokaryotic cells can be anaerobic and aerobic as well, which then makes to for prokaryotic cells that they are very flexible, they are very adaptive. Okay, we will talk, I think, yes, on the next slide about archaea. Archaea are something what belongs to prokaryotic cells, but they are not. They are definitely not bacteria, and we do not know a lot so far about archaeas because they were established as a individual living form of organism only in 1977. Nevertheless, we know that they are very adaptive and they can colonize also um, environments which are very extreme. For example, environments where is a really high temperature or low temperature, or which are very acidic and so on. And that's thankfully because of their metabolism. It can be also anaerobic, it can be aerobic, and they can adapt much more easily comparing to the other organisms or multicellular organisms as we are. Okay, we are very complex. We as humans are very complex. We have very specialized organs and cells. Nevertheless, for us, it would be really hard to live in an environment where there is a 95 degrees Celsius. Okay, we would die very instantly. Nevertheless, archaea very adaptive, and they can survive. They not even survive. They can evolve there. So thankfully, their metabolism. And endocytosis and exocytosis is one of the last differentiation factors. Endocytosis and exocytosis is also topic we will discuss in this chapter. Do you have any ideas what it can be? Endocyte? No. Okay. So basically, it's I hear my echo. Maybe who is unmuted? Or maybe just volume down. It will be also good. Okay, endocytosis and egg. I will check. I will try to fix it. Give me a second. Okay, now. Okay, perfect. Works. Okay, endocytosis and exocytosis is basically, I think it's best when you see the picture. Um, Okay, we will discuss this later. I need to zoom out. Okay, endocytosis. So there are some particles outside of the cell and how they get inside to the cell, and the exocytosis is exact opposite. So there is something you want to get out of the cell. So how you will get that out of the cell? Okay, endo and exocytosis. Endo and exocytosis. um, Endo and exocytosis uh, is not present in prokaryotic cells. However, it is present in eukaryotic cells. What is essential to mention that those processes, one more time, get me there, those processes require energy. It requires energy. You have to spend some amount of energy for doing that. Okay, it's not passive transport, it belongs to active transport. We will get there. Okay. Okay, guys. So let me get to the another point. What do you know, or guys, can I ask you, have you seen the video I uploaded about Archaea on YouTube? There was a video in Google Classroom. I uploaded it. Let me check. I have to. Okay. Okay, perfect. Yes. So have you checked that? Have you seen that video? Just okay, we will do a poll. Yes, I saw. No, I didn't. Okay. We will discuss it now. I'm just curious whether you are checking the resources or not. There is no wrong answer, okay? No wrong answer. Okay. Perfect. So here are the results. So most of you have seen that. Perfect, guys. So for those who are not, no matter, you can also check the video. It's very short. I think it's only 5 or 6 minutes, and you can check it after the session. I think it's created in a very interesting way. So, and it won't definitely waste your time, and it's short. So archaea versus bacteria. Archaea are let's discuss archaea. Archaea are very similar in size and shape to bacteria, and they are also unicellular. And since bacteria and archaea are two types of prokaryotes, uh, that brings us that all of the prokaryotes are unicellular. Um, so we have prokaryotes, bacteria, and archaea, and for eukaryotes are just eukaryotes. Okay, so there is just three groups basically, we can say. Some archaea are found in extreme environments, and for example, to those what I already mentioned, it can be hot springs, for example, hot springs, okay? Um, for example, but also not just in hot springs, in oceans, soils, and also inside the other organisms, also in humans. There are some types of archaeas which are able to be found also in humans. Uh, there is, as we already said, the shape and the structure is very similar to bacteria's. It has also a cell wall and flagella. Let me just remind you what is flagella. Flagella is that motion organ here. Yes, remember, and also cell wall. Perfect. However, what is different when we are talking about cell wall? One of the important substances is peptidoglycan. Peptidoglycan is essential for many of the cell walls in prokaryotes, but archaea doesn't include peptidoglycan. This is something when you are doing notes, please try to know that because this can also occur in an admission question because this is one of the main differences regarding the structure of cell wall in bacteria and in archaea. So there is, talking about archaea, there is no peptidoglycan. Okay, no peptidoglycan. Mhm. Yes. Okay, there is some also ester and lipid bilayer linkages, but you will see that in a video, and you already saw that in the video, is not so important as this peptidoglycan. Okay, so this peptidoglycan is very important for you. Yes, in addition, the flagella of the archaea work the same way as those of bacteria. However, they evolved from different structures. So they work similarly, but the origin of them is different. And what is also different is that some archaeas have does not have a lipid bilayer, but have only lipid monolayer. What it means? Are you familiar with the term lipid bilayer, lipid monolayer? No? Not so far. Okay. We will get there when we are talking about um, cell membrane. Okay. So I will just draw it here. Okay, guys. This is basically how our cell membrane look like, and this is also how does cell membrane in prokaryotic cells looks like. This is a lipid bilayer. Let me change to the pointer. You have two layers of phospholipids there. Phospholipids, it's one of the essential parts of cell membrane. If we have a lipid bilayer, phospholipid bilayer, it means we have two layers of it. Okay. Nevertheless, in archaea, some archaeas have that just one layer of it, and this is one of the factors which makes them so special, and also why they are so adaptive. It's also definitely there are other things uh, which can affect that, and it's still, let's say, a case of a survey and of a research, but currently it is known that lipid monolayer is more adaptive to extreme environments as lipid bilayer. So that's why archaeas can live in such extreme environments as they live. Okay, so this is one of the factors. Yes. And what's interesting currently or in the recent years is that Archaeas were till 1977 part of bacteria in prokaryotic cells. They then classified as then after 1977 they were classified as a separate group of prokaryotes, and currently these days uh, we know that there are more similarities between archaea and eukaryotic cells than with the bacteria. So it's very interesting that uh, 50 years ago they thought that archaea and bacteria are almost the same thing, and in 50 years of research they found out that there are less similarities with bacteria and more similarities with eukaryotic cells, so totally completely different group. Yeah, so if we get back, we can say that prokaryotic cells we have back and archaea, okay, since 1977. And before 1977 they thought that archaea are just as bacteria. After that, no, but still currently um, we divide prokaryotic cells in bacteria and archaea, and all eukaryotic cells are just eukaryotic cells. But we don't know, maybe in a future research, maybe in a really near future, archaea will be classified as a part of eukaryotic cells. We do not know what will upcoming years regarding the research give us, but it's very interesting, I think. Okay, guys, so that was a bacteria versus archaea. That was everything so far regarding the prokaryotic um, prokaryotic cells. Nevertheless, I'm checking now the time. We have only 10 minutes left. Mhm, mhm, mhm. How to handle that? Okay, guys. I will ask you because now we will get to the more interesting for you, more interesting part, which are eukaryotic cells. Um, as a future doctors, this is more interesting for you, definitely. My question is, have you seen the video regarding the cell? Uh, what is also in a Google Classroom? Yeah, you have seen that. Hmm? Most of you have seen that. Okay, there are.
Hey, sorry,
Yes, Sora, yeah, go on.
I didn't hear you. So please, one more time, please.
Yes, the question is if you have seen the video regarding the cell inside Google Classroom. There is a link.
Okay, thank you
Okay,
very much.
you're welcome, you're welcome. Okay. Mhm. Okay, so most of you have seen that. Perfect, guys. That's great. Um, okay. Uh, I will show you. I will show you what is a following activity. Breakout room activity. Basically, I intended to now distribute you into two pairs, into more pairs, and you can collaborate on each of those organelles. Nevertheless, we are running out of time. And what comes to my mind that we can have this can be your homework. I will tell you who is going to do what. You can reach yourself. It doesn't have to be long, really, like 2 or 3 minutes. Uh, you will speak about or prepare something which will last 2 minutes. Just introducing to the others those organelles. Is it okay for you if we will do it this way? Yes? Okay. Let me just check how to manage that and I will tell you who is going to do what. Okay? And you don't have to be bothered with that. Tomorrow we are going to cover all of those things. Okay, just let me check. Let me handle the. Okay, that makes sense then. Okay, guys. So please, I will now tell you who is going to do what. Okay. So Mona and Yuiko. Mona and Yuiko, you are going to do endoplasmic reticulum. Okay? Mona and Yuiko. Perfect. Mona and Yuiko. Yuiko, are you here? Or can you maybe just answer to the room chat?
Yes, okay.
Okay, perfect. So please reach with Mona, and you, both of you, will cover endoplasmic reticulum. Okay, let's just gather some knowledge, and you can prepare a very short presentation, maybe just with one slide, and for 2 minutes long maximum. Okay, perfect. Let me just check. Okay, then Golgi apparatus, Misato and Nao. Misato, Shimoda, Nao, Saburi. Okay? Okay for you guys? Perfect. Misato and Nao are together. Then Aoi and Satsuki. Aoi and Satsuki, you are going to have lysosomes. Okay? Aoi, are you here, Aoi? I can't see you. Yes, okay, I see you. Aoi and Satsuki. You are two are going to have this. Perfect. Okay. Then ribosomes, Shoko and Sora. Okay, Shoko and Sora. Okay for you.
Okay.
Perfect. Shoko and Sora, you are going to have ribosomes. And the last part, cytoskeleton and cytoplasm for Yuki and Yuta. Okay, Yuki and Yuta, you are going to have cytoskeleton and cytoplasm. Okay, guys. So just prepared something. You are not going to be graded, so you don't have to bother. Even just to create you and or start. I just want to, or the intention behind this, create an environment how you will be handled in medicine. Okay, so then you will have like some kind of task, and you will have to prepare something. You have to study something by yourself. Nevertheless, it will be. This is just really. I, as I said, as I wrote, I want to create a student-friendly environment. So you are not graded here, and I'm here for you to help. Nevertheless, this is some kind of activity which are from different group of learning. This is from active learning activity methods. So okay, ladies and gentlemen, that's everything. We can end the session here. So basically tomorrow we will start with the cell organelles. Okay, perfect. Um, okay, guys, do you have any questions towards me so far? No? Okay. If you do not have any questions, tomorrow, when we are done with the cell organelles and we will move to the transport, I will ask you maybe for feedback, because today we had the first part, the first almost the first session, whole the first session was about the technicalities and the introduction and stuff around. So then we will discuss, or you can tell me your feedback. However, if there is anything, any questions, anything you would like to maybe modify, don't hesitate, reach me via email, okay, or to Google Classroom. Okay, thank you very much for participation, that you were active. Thank you very much for that.
that you are not hesitating to ask questions, or you don't know something, or you just want to repeat something, please just continue in this style. That's great. And yes, I'm looking forward to see you tomorrow. Thank you guys. Thank you very much, and have a nice rest of the day. Bye. So you can leave now.
Thank you very much.
Thank you very much as well.
Thank you.
You're welcome.